Hi class, this is your lecture on chapter 7, which is on the endocrine system. We've got another chapter that we're going to cover, chapter 23 on endocrine pathologies. But first, let's learn about how the endocrine system works. We will be going through most of this chapter, so please read all of the pink highlighted sections of your textbook. All right, so first of all, let's begin by describing what a hormone is. What is it made out of? How long do they last? Well, hormones may be made out of one of two types of amino acids. They can be made out of proteins or peptides. And then they also can be made in the category of being a steroid. And this is important because steroids are fat soluble. They're derived from cholesterol. So we have the ability to have hormones go into all of the different compartments of the body since we have different molecular characteristics associated with our various types of hormones. Now, they do travel through the blood. They are not under paracrine or autocrine control mechanisms. Rather, they target a cell that has the right receptor. And they are able to do this at incredibly low concentrations. We're talking about 10 to the minus 9 to 10 to the minus 12. I mean, think about how many zeros is in front of that number there. So they are, and the reason they are able to work effectively at such low concentrations is because they target a cell specifically by binding to the right receptor. Now, hormones do have a half-life, and a half-life is the time for half of the molecule to decay or be broken down. So we do have different mechanisms that are at play that lead to the half-life or break down the hormone. So most of our hormones do fall into the category of being protein peptide hormones. And these are going to bind extracellular receptors. Our steroid hormones are based on cholesterol, and we'll look at an example of this structure in a moment. But this means that since they're all based on cholesterol, all of our steroid hormones do share a common structure and somewhat of a common function because of the structure that comes from the cholesterol molecule. So we spend a lot of time in medicine talking about the damaging effects of elevated cholesterol, the bad cholesterol. But here's an example of where we need hormones, or should I say, here's an example of where cholesterol is beneficial through the creation of our different steroid hormones. And then the amine hormones are based either on tyrosine or tryptophan. So these are the two amino acids that can be used as the basis of this group of hormones. Now, hormones have that half-life, and we have three main mechanisms that are at play to create or enforce this half-life. First, they can be degraded in the liver through mechanisms in the liver. They also can be filtered out through the kidney. So they would enter the filtrate. And then, as you know, if material is not reabsorbed out of the filtrate, it leaves the body in the form of urine. And then our third mechanism is that the target cell itself may have a mechanism to break down that hormone after it's utilized it. And that would turn off the pathway. Again, hormones are very effective at such tiny concentrations. We can't just leave them around, or else they would continue to have an effect because they would target the cells that have that receptor. So it's kind of like a magnet being on the cell that pulls everything right to it, which speaks to the low concentrations at which hormones are effective and functional. Now let's look at the example first of the peptide hormones, because the majority of our hormones are in this category. Now, they are made in the rough endoplasmic reticulum. So that's what the structure is right here that we're looking at. And even though it's not labeled as the rough endoplasmic reticulum in this figure, it's got a ribosome on it. So you know that means it is the rough ER. And part of the job of the rough ER is to synthesize hormones that are secreted from a cell. So actually any protein that is going to be secreted from the cell travels through the same pathway. Now, we begin with what is called a pre-pro hormone. The pre means one thing, the pro means another thing. The hormone is functional. So we've got two steps here of non-functionality with regard to the way the hormone functions outside of the cell that it's made in. But these are needed to send our hormone through the right cascade of events in the endomembrane system. So first of all, the pre part refers to the presence of this red signal sequence. And all the signal sequence does is it sends the pre-pro hormone forward in its pathway. Next, so now we're just a pro hormone. We've lost the signal sequence. That's what the pre means, the signal sequence. And I think of signal sequences just like a zip code. They just help to direct the pathway of travel. What happens next is the prohormone buds off of the rough endoplasmic reticulum in a transport vesicle. So you can see the color of the membrane around the transport vesicle matches the color of the endoplasmic reticulum because that's where it came from. Our transport vesicle contains it contains the prohormone, and it fuses with the membrane of the Golgi apparatus, which then delivers the prohormone into the Golgi complex. So the Golgi complex is like that giant post office for our cells. It's a molecular post office. So the I'm confusing myself here. The prohormone travels through the Golgi apparatus, and then it buds off of the shipping side of the Golgi apparatus in a transport secretory vessel. So it still is the prohormone while it's in the Golgi apparatus. It isn't until we're in the secretory vesicle that we get these peptide fragments cleaved off, and now it's an active hormone. So it begins as a pre-pro hormone, loses the signal sequence, becomes a prohormone, which goes through the Golgi apparatus. It buds off into the secretory vesicle where the peptides pieces are cut off. And now we have our active hormone that is released from the cell by exocytosis. And then the hormone travels across into the blood vessels and is transported to the cell that has the right receptor through the bloodstream. So our peptide protein hormones, they are going to bind extracellular receptors. Okay, so let's look at what happens once the hormone gets to the target cell. We just looked at how the hormone is made, but now we're going to see what happens. So the first reason why hormones are able to function so effectively at low concentrations is because they target cells specifically by having the matched receptor. So that's kind of the first criteria that creates that. So in this example, we have two different hormones shown by different shape, and notice that they have different receptors that they bind. Our square hormone binds the receptor with the square shape, where the diamond hormone binds the diamond shape receptor. So the hormones travel through the plasma and interstitial fluid until they get to their target receptor, and then they bind that receptor. They do not enter the cell. They never come inside. They instead initiate signal transduction, which is going to result in the activity of a protein that's inside this target cell. So in this case, with our square hormone, it binds to its receptor, which then activates the gate control of an ion channel in this example. Notice it also feeds into an amplifier enzyme, which is also in the membrane, but it's crossing over into the intracellular space too. The amplifier enzyme, like the name suggests, just makes the signal bigger. And it does this by activating a second messenger system, which then makes the signal even bigger. So we've got two mechanisms of amplification here. We've got our amplifier enzyme and then the second messenger system. In the case of our diamond hormone, it binds its correct receptor and it initiates signal transduction through a tyrosine kinase. Now, all tyrosine kinases do is they lead to the phosphorylation of something. And in this case, it activates a protein that is already made. So this protein inside the cell is stored inside this target cell, ready to go. It does not need to be transcribed and translated. That would take time. So part of the way that we get our very quick responses is because we've got two main mechanisms to amplify the signal to act on a protein that is already produced. So our secondary messenger systems are responsible for the speed, the quickness in which these responses occur. And again, we have to have that protein already made in order for it to happen quickly. Hopefully you appreciate by now the process of making a protein from start to finish. It's a pretty lengthy response. But if we're talking about a hormone, we don't have the time for that to happen. It's got to already be there in these cases. And then the proteins associated with whatever cellular response is coming from the cell that has been targeted. So again, peptide hormones do not enter the cell. They bind extracellular receptors that initiate signal transduction, resulting in the activation of a protein that's already been made, synthesized, and stored in this cell, in the target cell. Now let's talk about the steroid hormones. Now they all are derived from cholesterol. So we spend a lot of time in medicine talking about the unhealthy effects of bad cholesterol, but this is an example of a needed effect from cholesterol. We need it to make our steroid hormones. Now, steroid hormones are made in very few places. They are made in the adrenal cortex, which is the outer part of the adrenal glands, which sit on the kidneys, or the gonads. And if we're talking about male versus female, there's different hormones that come from the different type of gonad. Now, these hormones are made as needed. They are not stored. That is a big difference between the protein peptide hormones versus steroid hormones. Protein peptide hormones are stored and can activate those systems quickly, where our steroids are made on demand. They too are transported in the blood, but hopefully that sends a little bit of a red flag up for you, because we're talking about cholesterol and we're talking about steroids. Cholesterol is a lipid. It is not water soluble. So how on earth do these steroid hormones move through the bloodstream then? Because, right, our bloodstream is not lipid soluble, it's water soluble. They do this by binding what's called a carrier protein. So it's like a bus, but it's made out of a protein and it shuttles the steroid so that it can move freely through the bloodstream. Now, steroid hormones have a considerably longer half-life than do the peptide hormones. So this is an example, like we've seen many times before, where we have a range of effects that cover different time frames and also allow the access to different parts of the body, specifically a slow response versus a fast response, or a response in the bloodstream versus somewhere in a lipid soluble compartment of the body. So let's take a look and see what some of these actions are of our steroid hormones. We'll study some of these also in the next chapter we do. All right, I know this is a really complicated, probably scary looking figure, but it shows you the relatedness of all of these steroid hormones that are based on cholesterol. So this is the structure of cholesterol. So that means that every single steroid hormone has this structure in it. So what makes them different are the side chains that attach onto the cholesterol molecule. It's not the cholesterol itself, because they all have cholesterol. That's kind of like saying an amino acid is unique because each amino acid has an amino group and each amino acid has a carboxyl group. Well, that's not what makes them unique at all, because every amino acid has those parts. What makes the amino acids different from one another are the different R groups. Here it's the different side chains. So we begin with cholesterol. We've got an intermediate that then is converted to a hormone called progesterone. And this is produced mostly by the ovaries, but some of it's produced by the adrenal glands too. Progesterone can act through the action of an enzyme to be converted into corticosteroid, which then is converted through another enzyme into aldosterone. And we learned about what aldosterone does in the renal system with regard to sodium and potassium balance. So this is the structure of aldosterone, but notice it's got all 4 rings of the cholesterol. Our side chains are different. And here's just a picture showing the adrenal gland sitting on top of the kidney. So we have 2 adrenal glands. Progesterone can also follow another pathway where it is converted into testosterone, and then into, furthermore, another hormone, dihydrotestosterone. So testosterone is converted into estradiol, and this is through an enzyme. So these are our male versus our female hormones, but clearly we have very different phenotypes between the male and female biological expression. I'm trying to think of an inclusive way to say that. I apologize if I didn't say it in the best way. But just simple changes in these side chains. Here's cortisol. We're going to spend a lot of time talking about cortisol because it's a really important hormone that is associated with a pathway leading to the action of other effects, as we'll see. So each of these steps that has a star here requires a different enzyme, but I'm just not showing you them all. It's just complicated to list them all, and you don't need to know these. What I'm interested in you learning is that our steroid hormones are all derived from cholesterol. Now, these are made not in the rough endoplasmic reticulum. This is not protein. Steroid hormones are made in the smooth endoplasmic reticulum of the gonads and the adrenal cortex cells. They aren't stored. They are made on demand because cholesterol is already made. So these other changes aren't as time consuming or as significant as is the creation of cholesterol. We've already got our precursor molecule ready to go. Now let's look at how our steroid hormones interact with their target cell. Now, our steroid hormones are bound to a protein carrier. So the steroid is the yellow circle, and then we've got this protein carrier, and that enables it to move through the water-soluble environment of the blood. The steroid disassociates from its carrier, and then it crosses the membranes, the different surfaces, and we've got a couple options. Some steroids are able to bind to cell surface receptors, and this leads to a very rapid response. They can also bind to receptors that are in the cytoplasm, and then that will lead to transcription. They can also bind to a receptor that's in the nucleus, and that too is associated with transcription. So we've got a receptor hormone complex here that's binding to the DNA, and this is working similarly to a transcription factor, where it either activates a gene or turns off or suppresses another gene or even groups of genes. It does result in the transcription of messenger RNA, and then that moves out through a nuclear pore to the cytoplasm, where it then goes to the rough endoplasmic reticulum so that it can complete its process of translation, resulting in the production of a new protein that is produced in the target cell. So big differences from our protein peptide category. The protein peptide hormones, they are activating an existing protein that's already made inside the cell. They are quicker. Our steroid hormones result in the production of a new protein. So that takes time. And then we've got the differences in terms of where the receptors are located. Now, our third category is the amino acid-based hormones, and they're made from one of two amino acids. So tryptophan is one, and they are made in a place on our body called the pineal gland, and they're made, melatonin is an example. The second amino acid that is utilized as the base of this group of hormones is tyrosine. And this is where the catecholamines come from. We've learned about our catecholamines already. So these are molecules including epinephrine, norepinephrine, and dopamine. Now, the catecholamines behave similarly to the peptide hormones in terms of how they move and how they interact with with receptors and their target cell. The thyroid hormones are made from two tyrosine molecules. So there's two tyrosines in them to give us our thyroid hormones. They behave like steroid hormones. So our catecholamines are going to travel and function like the peptide ones, where the tyrosine, the two tyrosine based molecules more behave like a steroid. So this is tyrosine up top. And then on the left side in this pink color box are the catecholamines. And all that's different here is the side chain attached to tyrosine. So we've got the difference between dopamine and norepinephrine is that we've got an OH here and an H here. And then in epinephrine, we have a methyl group on the terminal nitrogen, whereas there's a hydrogen on the other two catecholamines. The thyroid hormones, we've got two tyrosines, and they have iodine on them. Iodine, and this is partly is it allows for, in the case of a person having thyroid cancer, the treatment can actually be targeted to go right to the thyroid and spare the rest of the body because of the affinity for iodine with the thyroid. So down at the bottom, we're looking at, so this is thyroxine, which is called T4 because it's got 4 iodine molecules on it, 2 on each of the tyrosines. And then this bottom one is called thyroxine, which is T3 because it's only got 3 iodine molecules associated with it. So how is all of this controlled? Well, the endocrine cell is able to perceive the stimulus by interaction with the receptor, and then it can respond. So we call this trophic. And what this means is that one hormone regulates the release of another hormone. And we'll look at a cascade of hormones that are trophic. We also have, the nervous system can also stimulate the release of hormones. These would be properly called neurohormones when they're stimulated by the brain, when the brain produces them. An example would be the hypothalamus of the brain that's responsible for producing and controlling the release of hormones. We also are able to, the neurons are able to secrete some of these neurohormones. For example, epinephrine, norepinephrine, and dopamine are made in the medulla of the adrenal glands. So it still is considered a neurohormone, even though it's not being made in the brain, because it is under the control of a pathway that originates from the brain. We'll see. So trophic hormones, again, are peptide-based hormones that influence the release of another hormone. The hypophysiotrophic hormones, that's a mouthful. These are peptide hormones, and they're produced in the hypothalamus. So that's where the prefix hypo comes from here. It means hypothalamus. And then these hormones release other hormones from the pituitary gland, the anterior portion of the pituitary gland. Okay, so this is looking at the structure of the pituitary gland. So it's right here. It's actually 2 glands that are made out of completely different material. So we've got different embryonic tissue even that's associated with these different regions of the pituitary gland. And it's protected by the bone of the skull. So anterior would be towards the front, posterior would be towards the back. So zooming in on the pituitary gland, we can perhaps better see the cartoon representation of this different tissue. So this part here is the posterior behind, and then this is the anterior pituitary. So the anterior pituitary section is a true endocrine gland because it's made from epithelial tissue, whereas the posterior pituitary gland is made from the neural tissue, and then it's protected within this bone here. Okay, so now let's look at the hormones, begin our discussion of the hormones that are from the anterior pituitary gland. There are 6 of them, 6 hormones, and they all fall into the category of neurohormones because they're under the control of the hypothalamus, which is part of our brain. So here we have our hypothalamus synthesizing we have our hypothalamus synthesizing neurohormones here, and they travel through a portal system, just like we've learned before, 2 different capillary beds that are connected through blood vessels. So here's one capillary bed, and then here's the second. So these neurohormones are released into the capillaries, and they go directly down to the anterior pituitary gland, where they are then able to act on those endocrine cells that are present in the gland. And the types of hormones are shown here. We're going to go through them in the next couple slides. So it is the cells of the anterior pituitary gland that are responsible for producing all of these different hormones. And we're talking about effects on the musculoskeletal system, on the mammary glands, the thyroid, the adrenal glands, as well as the gonads. So here's our names of the hormones that are released from the anterior pituitary gland. So we've got prolactin, we've got growth hormone, thyroid stimulating hormone, adrenocorticotropic hormone, luteinizing hormone, and then follicle stimulating hormone as well. Okay, so here's a table that summarizes, again, those 6 types of hormones that are from the anterior pituitary gland, as well as giving you a couple examples of how they're inhibited. So let's go through the main characteristics. So prolactin is important in lactation in the female, and it's an inhibitor. Excuse me, I can't talk today. It is a inhibitory hormone. I'm gonna back us up. So prolactin is in it. We've got dopamine, which inhibits that activity. For growth hormone, this is associated with metabolism, and we're talking about production, stimulates hormone production from the liver, and this is all tied to metabolism. We have 2 examples of the gonadotropins. So there's follicle stimulating hormone and then the luteinizing hormone. And these are associated with hormones that are in the ovaries or the testes. Thyroid stimulating hormone is in control of the hormones that are secreted and produced by the thyroid gland. So those are those amine-based hormones. And then the adrenocorticotropic hormone is responsible for controlling the production and synthesis of hormones that come from the adrenal cortex, specifically cortisol. Now, the posterior pituitary gland is associated with 2 hormones. So the anterior is with 6, the posterior is 2. And one of these we've already learned about, it's vasopressin, which has a role in the kidneys, as we learned about in our renal section. The second hormone associated with the posterior pituitary gland is oxytocin. And oxytocin has actually been studied a lot recently. We know about its roles in, it's called the love hormone, because of it stimulates bonding between mother and child. So one of the ways that oxytocin is released is it can be induced to be released is by having the baby lay on the chest of the mother or the father. Fathers can make oxytocin too, or males can produce oxytocin too. But oxytocin has some other really interesting effects. And this was something I studied at the University of Arizona. It's associated with preventing and getting rid of inflammation. So we had a study called the HUG study. And it was a study that was done in subjects that had rheumatoid arthritis, which is an autoimmune disease. And they had pretty significant rheumatoid arthritis. They were considered to have moderate to severe disease. And what we did in this study is we categorize the subjects based upon the amount of hugs they received. I know it sounds weird. So we had one group of patients that had a partner. Doesn't matter who that partner is. It can be same sex, it can be opposite sex, it could even be a dog or a cat, we came to find out. But the point is that ventral to ventral surface being in contact with each other leads to the production of oxytocin. And we saw in our rheumatoid arthritis population that the patients that had hugs produced more oxytocin than those that didn't. And furthermore, their inflammatory markers, which correlate with the disease process, decreased. So we recommended that people hug each other, and it works with dogs. The key is that you want about 30 seconds of contact between whoever you're hugging. So if you've got some inflammation or you're feeling down, go hug somebody. There's a lot of good data out there now to show that it has really powerful effects other than just what we think about with mothers and labor and milk production. So the posterior pituitary is back here, and it's made out of that neural tissue. It's not a true endocrine gland, but it releases, the hormone is packaged in a vesicle, and then it moves down through the blood vessels into the posterior pituitary gland, where it's then released and travels over to the bloodstream, where it then travels through the rest of the body. So this is for you to think about, and I did put the answers in the note section of the slide, so you can double check your work by clicking the note view, and then you'll see the answers right below. But we've got our two hormones that are made by the posterior pituitary gland. What are their targets and what are their functions? All right, so let's talk a little bit more about the hypothalamus and the anterior pituitary gland. So we've got this portal system that we were looking at, the hypothalamic hypophyseal portal system. And this ties us into that property of trophic hormones, where one hormone is associated with regulating the release of another. So first of all, we get it released into the first capillary bed in the portal system, and then it goes through the portal vein, and then we have release into the second capillary bed in the portal system, and that allows it to travel to the target tissues through the bloodstream. And this ties to those very small concentrations of hormones that are effective because they bind to a target cell through a receptor. So it's only acting on cells that have that right receptor. Okay, so this is the whole pathway associated with the hypophysiotrophic hormones. So we have our stimulus that acts upon the hypothalamus. The hypothalamus produces hormone 1, which acts on the anterior pituitary gland to produce hormone 2, which then acts on the endocrine gland part of the cascade to produce hormone 3, which then acts on the target tissue, leading to the response. We do have 3 different feedback loops, negative feedback loops that can turn off different sections of this pathway depending upon how far we are in it. So we have one that begins, is initiated by the production of this third hormone that can feed back up to either the pituitary gland or the hypothalamus itself to turn off the pathway once enough of that hormone is available. Those are both considered the long loop negative feedback control mechanisms. And then we have a short loop one that goes from the hormone 2 back up to the hypothalamus. So what I would like you to do is put in the hormones here. name the hormones so that you know the pathway. Here we go. So we've got the hypothalamus, which leads to the release of corticotropin releasing hormone. That's hormone 1. That acts on the anterior pituitary gland to release our second hormone, which is the adrenocorticotropic hormone, which acts on the adrenal cortex to produce cortisol. So that's hormone 3. And cortisol is what travels to the target cells and tissue and binds to its matched receptor, leading to the cellular response. So then we've got our long feedback all the way back, and then our shorter feedback mechanisms. So this is the hypophysiotropic hormone pathway that results in the production of cortisol, or the secretion of cortisol. So try to think about where that short loop negative feedback would go from, right? We're just looking at both long loops. The short loop is associated with hormone number 2 in this pathway, ACTH. So in an endocrine stimulus response situation where we have 3 hormones, which hormones would be trophic and what is their source? So I want you to write this out, please. It will help you to get these concepts in your mind. It also will help you to have some resources available when it comes time to take your last exam, your exam 4. Some other questions to think about. We've got in the long loop negative feedback, which hormones inhibit the release of others? So it's important to know the cascade of events and what connects what with which negative feedback loop. So I'll let you fill this in. Again, you can check your answers by looking in the note view. Now, hormones are able to interact through different mechanisms. We have synergism, and synergy is an interesting concept. I'm just going to give you a generic example. So in an additive effect, 1 + 1 = 2. In synergy, 1 + 1 = 3 or 5. So what synergy is when 2 hormones act together, and together it causes a greater effect than could come from either of those 2 effects added together. There's also permissiveness, and this means that one hormone is not able to fully work unless another hormone's present. So the thyroid hormone has a permissive effect on the sex hormones. And then finally, we have antagonism, and this is when 2 hormones have opposite effects. I want to point out this is not equal all the time to competitive inhibition, like we learned about earlier in the semester when we were doing our intercellular communication. We'll look at some examples as we finish up this topic on the endocrine system and pathologies. So here's an example of synergism. So we're looking at time on the x-axis versus blood glucose level on the y-axis. And we've got cortisol, we've got glucagon, and we've got epinephrine. And we see their effect on blood glucose level individually in these 3 graphs down at the bottom. But when glucagon is added with epinephrine, we get a bigger response. And it's higher than if we were to take 100 plus 125, it should be higher. But look what happens when we add in cortisol. This is where the synergy really takes off. So now we're way above the effects of all 3 of these added together. That's what synergy is. It gives us a bigger bang for the buck by combining 2 or more things together. This also is a conservation in a sense, in that we don't have to have a 4th hormone created and regulated to do this. We can just add together, synergize together some of the existing hormones. All right, that finishes us for chapter 7. Pretty quick lecture there. And then I'll be back in a moment to go over endocrine pathologies, which wraps us up for the semester. I hope you're well. I hope your families are safe, and I'll see you soon. Bye.